Hello, everyone. Sorry, we started a little late because there was a line to get into the parking lot. Welcome to our Dean's Distinguished Lecture. Just as a little bit of background, we started this series back in 2014 as a way to invite the local community to learn about the kind of research that's being conducted here at UC Irvine. And we try to hold this lecture on a quarterly basis. And we generally invite faculty from the School of Biological Sciences, but periodically we reach out across the campus. And this year, to kick us off in 2026, have the distinct pleasure of inviting Dr. Tallie Z. Baram, who is a distinguished professor. She's actually the D. Shepard and Bren Distinguished Professor at UC Irvine, and she has appointments in pediatrics, anatomy and neurobiology, as well as in neurology. There are distinguished professors, and then there are distinguished professors, and Tallie is definitely in that latter category. She is an outstanding basic researcher working on developmental neurobiology and early childhood experiences. She has won many accolades for her research, and she's someone who I have had the pleasure of knowing for a long time. And so I thought I would share with you how I got to know Tallie, because it's a little bit of an unusual story. And shortly after moving here to Irvine in 1995, my older daughter developed this disorder, which actually sounds a lot worse than what it was. It was called partial complex epilepsy. And so we were very scared, and we went to a neurologist in Orange, and I don't remember who this was, but the guy looked at our forms, and he saw we were from Irvine, and the first words out of his mouth were, "What's the matter? Tallie doesn't accept your insurance." And so luckily for us, it was November, and which was open enrollment season, so we immediately switched our insurance to the one that Tallie had accepted, and she took us on as a patient. And I have to say, to this day, I tell everyone, I have never met a physician who has better bedside manner than Tallie Baram. We were scared, we didn't know what was going on, and she talked to us so intelligently and so calmly and walked us through the entire process. And within, I think, a year and a half, 2 years, Caroline outgrew it. But that experience stayed with her and motivated her, because it really made her want to become a pediatrician herself, and she did. And so she's a med ped specialist over at Loma Linda, and she always tells the story of how this pediatrician impacted her life, and she wanted to have the same impact on other people's lives. So Tallie, your reach across this campus is just absolutely just expansive. I think she embodies what I like to say: there are leaders that require a title, and there are leaders who don't have a title. And Tallie is a leader that doesn't have a title. There's hardly any major decisions in the medical school or on this campus that are not made in which Tallie's hands are not involved. She is someone that we often go to strategize with. She was absolutely instrumental in helping us launch the Depression Center and getting Diego Pizzagalli here as our director of that center. So let's give Tallie a warm applause.
Well, I don't know what he was talking about. But it really is a pleasure and an absolute privilege to be here with you. And really, I'm grateful for Dean LaFurla, and actually for the school and for the university to allow me to be part of you. And I think the one thing that motivates all of us is really to ask important questions and to really try to answer them. And one of the most important questions, I think, is who are we? What gets us to be where we are? And this is really what we're going to be tackling right now and addressing, like how things that happen to us early in life actually determine or help determine who we end up, how we feel, how we think, and what our behaviors are. So we really have a challenge which we need to address, and that is to understand really what is the impact, if any, of things that happen to us really early in life. And we need to know that not at the population level, but we really need to know each one of us, why are we who we are, and how were we influenced by what happened to us. So we will address that really by asking several questions. First, how does a brain really remember and code what happened to us early in life? And we'll focus particularly on early life stress or early life adversity that I will sometimes refer to as ELA. The second question is, okay, things that happen to us early in life happen there transiently and then they quit. But we know the effects of them like 40 years later. So how does that work? How do you transform something that's transient into a lasting impact? And finally, the most important question that we'd like to know is, okay, we can go do research in the lab and find all kinds of discoveries, but how are these discoveries relevant to us, to our kids? And where do we go from here? Okay? So that's the game plan. So let's start off maybe with what we do know. Well, we do know that our cognitive and emotional well-being and function are largely driven by genetics. Genes that we got from our parents, from our family, that has got a huge contribution to who we are, and twin studies and many others have shown that. But we also know that things that happen to us, influence from the environment, particularly early in life when our brain is so malleable and sensitive and plastic, really have a major impact. So this is what we know. We also know, when we go narrow our focus to adversity or stress, that there is a very large body of work, epidemiological and many different studies, suggesting that early life stress or adversity of many different kinds, poverty, extreme hunger, migration and war, and maternal depression and others, all do have an impact. Because if you look at large populations of people, this type of events is really a risk factor for later life problems with emotions and even mental illness, and also a lower cognitive function throughout life. Now, this is not trivial, because when we think about the overall burden of early life adversity, only about 42% of the world's children don't have any adversity. But people have developed scales to assess stress or adversity, and you can talk about 1, maybe poverty, or 2, maybe poverty and neglect, and so on and so forth. And you can see that a large proportion of the world's population of children really do have adversity. So I think it's kind of important to figure out, really, is adversity really lead to a problem later in life? Because when we have statistical association, that does not mean that one causes the other. There may be very well a coincidence. I mean, maybe most people in California have brown skin, but is this because of suntan, or do they come from Hispanic origin? So a lot of possibilities that we need to address. And this is particularly important because although it's sort of a truism that early life adversity is associated with maybe less than optimal outcome, there are some really good studies suggesting that that may not be the case. And here we need to really think about the difference between population level and the individual level, which is what we're all interested in. So let me carry you through this. This is a paper that looked at the world's largest prospective studies of early life adversity. One is from New Zealand, from Dunedin, and the other one. And what you see here on the y-axis is the prevalence of any mental health problem or physical health problem. And what you see on the bottom side is whether the children had 0 adversities or 1 or more and whatever. And what you see here, it's a no-brainer. The more adversities you have, the higher the probability that you will have either a mental health issue or physical health issue, right? Done deal. Well, it really isn't, because this is at population level. But what we really want to know is if Giulio and Maria had the same type of early life adversity, are they going to come out okay? And when you try to do that for an individual child, you get something like that. Now, don't get alarmed. This is actually something called a receiver-operator curve. And only about 3 or 4 years ago, I figured out what is the origin of receiver-operator, because Gary Lynch explained it to me. That actually started in World War II. You had soldiers in the United States and British soldiers sitting in the middle of the night in front of receivers, looking at radar blips and trying to figure out if these German rockets are going to hit or not going to hit. So receiver-operator curve. And if they would like to really ascertain that that's going to hit and they have to do something, the curve should look like the green line with specificity and sensitivity. But what you can see is that that important study, when you went to individual level, actually looked almost like a purple line, which is chance. And what that tells us, that Giulio and Maria can have the same early life environment, and Giulio can do fine and Maria can be a train wreck, and we don't have a clue how to predict what will happen to them. So we have a problem. Okay? In fact, what he tells us that what we think we're measuring as early life adversity really leave unexplained a lot of our ability to predict outcome. And it's important to predict outcome. We have limited resources. We'd like to try to convey them, to give them to people, to kids who need them, to try to prevent, to intervene, to prevent. We can know nothing. So, does early life adversity really lead to cognitive and mental health problems? Well, I submit to you that it's important to know because we'd like to know what the mechanisms are. We'd like to be able to translate it, as we talked about. But we really can't answer that in kids because nobody can ethically put early life adversity on top of a child. So we have to go to the lab. And my group has created a model of early life adversity, which is essentially simulated poverty. If you have a mouse or rat in a cage, they have two types of assets. They have a bunch of bedding, nice fluffy corn cob, and then they have some cotton wool type thing to build little nests. And we limited the amount of both the nesting and the bedding material. We call it limited bedding and nesting, or LBN. And what we do is artificially and randomly assign mice or rats to go either to one environment or another. We can actually cross-foster, so we throw out the genetics, and then we keep them there only for 1 week. 1 week only, okay? Postnatal day 2 to 9. And then we ask, okay, we have a control situation. Does early life adversity in these cages actually have mental health, quote unquote, or behavioral differences? And we thought the answer would be no, and boy, were we wrong. Just 1 week of that, and then you look at the animals as adults, have a bunch of cognitive problems that we're not going to deal with today. But there's also, quote unquote, emotional problems to the degree that you can assign emotion to animals, and that's a different discussion. But when we test the behavior, what we see that the behaviors are really, really changed. Males seem to be miserable. Something called anhedonia, the opposite of hedonism. They don't really care, they don't seem to have pleasure either in palatable food or sex or what have you. And in the females, in the adult females with a history of early life adversity, they seem to have increased motivation or craving for comfort food, for sex cues, and not only for natural rewards, but stuff that we did together with Steve Mahler, even for opioids, for drugs. So let me show you what that looks like. So we have many different tests because one test never really gives us the full answer. And we can see here that if you expose, in this case it's male mice, to palatable food, assuming you think Cocoa Pebbles is a palatable food, but that's a different story, they really don't care for it very much. You then expose them to sex cues. Now, I don't really show them Chanel No. 5. What we really expose them to is the scent of a female in heat. But again, they don't care. And sucrose, the same thing. For the ladies with a history of early life adversity, the lady mice, you see the opposite. They seem to like palatable food more, and this is particularly prominent when they have to work for it. They will work really, really hard. The motivation is the main thing that's affected, and also for sex cues. So 1 week in early life adversity in rats and mice directly, causally, can change adult behavior. So I think that is really what the take-home message here now. And of course, the next question is, how the heck does that work? So before we even can get to that, we need to try to realize, so we put them in cages, and the mothers were stressed, and they were stressed, and things change. But how did they get into the brain? I mean, clearly behaviors originate in the brain. How did it even get there? That turned out to be quite a challenge. Because now, the only thing you need to see in this slide is it's too complicated to understand. That's exactly the message, so don't even bother. But what we see here is that we have mice and we have humans, and we're trying to jump and apply things in terms of development between a mouse and a human, and that's actually not possible. It's not possible because the brain develops in modules and circuits, and they all develop at different times and different velocities, And the memory module, which is right here, hippocampus and amygdala, whatever, they all develop a different thing. So it's really not possible to exactly say that a baby mouse is the same as a baby human or whatever. But we do know one thing. When we think about the memory system, we know exactly when it develops in both mice and people. In mice, they really only develop somewhere starting on the 3rd and 4th week of life. And in kids, actually, do you remember what happened to you when you're 6 months old? Anybody here in a room remembers what happened to them when they're 9 months old? Please lift your hand. I don't see any hands because we don't. Somehow, prior to about 2 years, depending on what kind of memory you look at, we, as kids, are not able to form or store autobiographical events, memories, and we call them later in life. This is well known and is known in infantile amnesia, and it's really kind of crazy because we don't remember these things, but they clearly were encoded in our brain because they change us. And the one human-controlled trial about the Romanian orphans actually showed that what happened in the first 2 years of life is critical for adult behavior throughout life. So how does that work? I mean, normally if I would like to try to find out where does early life adversity get encoded, I would say, oh, in the hippocampus. But the hippocampus is not really functional. That system is not operational in the first couple of years of life in a kid and in the first few weeks of life in a mouse. So where in the brain do we even begin to look? And that was quite an issue, except modern technology. You want to find more stars? Go build a better telescope, okay? And of course you can. You build a Hubble, and you can see amazing galaxies and stars. Same thing has happened with brain science. We now have amazingly novel set of methodologies, and what we did is we used something called genetic tagging to really see what part of the brain is involved. We're not going to go into the details. I'm happy to share that with the cognoscenti, but essentially we can actually ask when cells are active in response to adversity or regular experience, and at a given time frame, maybe in postnatal day 5 or 6, we can freeze that picture. We can then go ahead and look at the brain much later in life and actually have visual colors, like the red here, that tell us where cells were active. And when we looked at the mouse brain at that, we saw that what was active here is the sausage. This slice is being done here, so it's kind of vertical, and this is much more posterior. And this sausage, which is the paraventricular nucleus of the thalamus, is really what lit up. And that was kind of crazy because most people, even neuroscientists, don't know very much about the PVT or paraventricular nucleus, and it was really unknown in the human literature to a large degree. But that, what was active? Well, it was really not only active in both control and early life adversity cages, but as you can see, it's massively more active in response to adversity. So that is a part of the brain that can tell the difference. In fact, it was the only part of the brain that could tell the difference among all the ones that we tested. First of all, I didn't really believe in this fancy technique, so I measured another technique, and it shows exactly the same thing. And then we looked at many other parts of the brain, and stress-related and fear-related and reward-related regions were also activated, but nothing else, no other part of the brain actually distinguished between the two. So we were kind of stuck with this PVT. So what the heck is it? Well, it's this elongated sausage that seemed to be a hub. It's connected to something called the nucleus accumbens, which is a hub of reward, and we'll talk about that in a minute, to internal states that tells us what goes on inside the body, to fear, to decision-making. It's amazing. It really is a hub. And because absolutely nothing was known in human PVT, we teamed up with imagers, particularly Mike Yasser and his group, and they were able to actually go look at some really high-power MRIs and really look at the functional connectivity of the PVT in the human. The good news, functional connectivity in human and mouse are very, very similar. So, okay, we have this strange brain region, the PVT, encoding early life adversity before the hippocampus comes on board. But so what? I mean, does it have anything to do with the altered behaviors that we see? I mean, is it possible that the PVT cells that are active are actually also the ones that execute or govern what happens early in life? And the answer here is yes, and this is like hot off the press. If you remember, the male mice that have had the early life adversity, or ELA, really was sort of miserable and anhedonic. And we were able to use the same type of technology that allows us to visualize the activated cells, actually also to silence them, to shut them up. So when we shut them up, we found some really interesting stuff. So let me walk you through that. If we take the same cells that are activated in control mice and then give a vehicle to these cells, nothing much happened in control mice. When we shut them down using a designer receptor type technology called DREAD, nothing happens also. If we look at the early life adversity anhedonic adult mice now, and we give them vehicle, they remain anhedonic. They consume a lot less than the control. But if we shut down the same cells that were activated early in life, somehow these mice have restored reward. And we've done the same thing in females, and it looks like the PVT really is an important brain region in the context. So we've making some progress in understanding how early life experience influence our adult behavior, at least in experimental model. And we discussed about how the brain can encode them. I'm sure it's not only the PVT, but this is what we know now. But then how does these early life events that happen, again, to us, maybe at age 2 or 3 or 4, everybody in a room, I think, can know something that influences them forever. And how does that get transformed to a persistent, to enduring, to something that's with us throughout the whole life, even if we can't remember it? So that, I think, really has to do with understanding sensitive period. So as we go through life, each circuit, each model, each part of our brain, and potentially also body, goes through a period where it is particularly vulnerable and sensitive. In fact, there's probably more than one. I mean, infancy and early childhood is what I showed you. We all know that adolescence may also be a very sensitive period for other things, such as perhaps social behavior, okay? And during that time, there is oftentimes a spurt of growth, but also a period of vulnerability, the mechanism of which are not fully understood. But one thing that happens at that time is that things that are transient are transferred or conveyed or transformed into permanence. How does that work? Well, there are two major ways in which it can work. One is through what is known as epigenetics. That is the idea that every cell in our brain functions because it makes particular genes and proteins and molecules at the right place at the right time. And that can be changed in sensitive periods. We're working really hard on that. I'm not going to tell you anything about it today because it will be here until midnight. The other possibility is the wiring. We have 100 trillion brain cells, neurons. In order for them to work together, they must be wired, and they are wired very carefully, like outstanding circuits that don't ever get jammed, and so that I have to let my computer down. Okay, that's a different story. So one thing that happens is that these circuits really function in a coordinated manner to allow us to have complex behaviors, to allow us to love, to hate, to think, to talk, to give lectures. And in a sensitive period, these, and early in life in general, these circuits are immature. Neurons come together. We have the fundamental machinery, but the synapses, the connections between the neurons, can either persist and create beautiful highways, or if they're not necessary, they can kind of wilt away and get pruned away. And one of the possibilities is that that may be what's going on with early life adversity. And we looked at that, particularly in an area of the brain that is highly involved in reward. Again, I told you we have circuits. One of the circuits is a reward circuit. And the hub, Dallas Airport, when it's not iced in, is really the nucleus accumbens. So what we elected to do is to see other circuits, particularly of projections or highways that go into Dallas and, that go into the nucleus accumbens, that are particularly stress sensitive, okay? That by definition. And we mapped and found a new one. And the reason that was stress sensitive is because that those cells express a molecule that is known throughout evolution to be a mediator of stress. And that molecule is called corticotropin-releasing hormone, or CRH. So what we've done here is, thanks to Matt Burney in the lab and others, we were able to find a completely new pathway, a completely new road, a completely new projection in the brain, a part of the circuit that was never known before. And I'd love to bring that to your attention because this is something AI will never do. There's no information that you can use large language or whatever to actually deduce that because it wasn't there. This is something that you need to do through empiric experiments to discover something that's completely new. And this projection not only was new, but it was actually kind of important. Because when we discovered it, we said, okay, this is a stress projection going directly to the reward hub. So what does it do? Well, again, we use this viral genetic methodology to stimulate it. And what we did, we took regular control male mice and stimulated using something called chemogenetic again, stimulated the activity of the projection. And what you can see here now, because we've already seen this before, it made those mice miserable, anhedonic. Essentially, they didn't care for palatable food, they didn't care for the scent of a female in heat, they didn't care very much for sucrose. In fact, if you remember, we got exactly, by stimulating the projection, what early life adversity did. And that got us to think, aha, maybe this projection is really influenced by early life stress and is therefore hyperactive and not quite wired right. Because when you think about this wiring diagram, which is a complex computer that is our brain, everything has to be just right. The difference between mental illness and typical individual is not very large. It's all a question of balance and really fine wiring. So what we did then is actually shut it down, again using the same technology. And surprise, surprise, when we shut down in animals that were miserable or anhedonic because they had a history of early life adversity, we were able to restore their typical reward behavior. Okay? So that was really interesting in terms of mechanism in the brain, in specific brain circuits that are influenced by early life adversity. We have here a projection, we have here a circuit that is really altered. So how can it happen? I mean, yes, you have early life adversity, you encode it in some of the brain, the circuit's conveyed elsewhere, and then all of a sudden you have a projection that's hyperactive, too excitable. Well, the way it works, again, to remind you that those circuits are actually very malleable early in life. The synapses can go either strengthening, maybe too much strengthening, or appropriately pruned. And that pruning is actually done by microglia. These are the immune cells that are resident in the brain, and they sit right around the neurons and kind of survey them. In fact, this is a better way to see that. The red cells are neurons, regular cells. The green cells are these microglia, and you can see them sending processes and surveying and kind of keeping an eye out on what's going on. They see synapses that don't belong there, they prune them. And what we found is that indeed early life adversity, probably through other stress hormones, influences the activity of the microglia. They don't do their job, they leave too many synapses, and therefore we have an hyperactive projection. If you want to read more about that, because I'm giving you just a smidgen, a taste, this is published by an incredibly brilliant former postdoc who's now running her own lab. So we're making progress. We now have also an understanding, both in epigenetic and particularly in fine-tuning, refining, maturation of brain circuits, how we can take transient events early in life and convey them into something that's permanent, because those changes in a circuit are there pretty much forever. But the question is, how is that relevant to the question that we originally asked about the relation of adversity to an individual outcome? Remember, we talked about this receiving operating curve? So we needed to really think if, again, as we said here, is it possible that we're missing? Is it possible that we as outsiders, as adults, when we look at the developing brain, we think we know what's not good for that brain, but maybe that brain doesn't agree with us. So there are many different aspects that we talked about. We talked about sensitive period. We talked a little bit about modality. Is neglect the same as trauma, the same as deprivation, the same as abuse? There's a whole literature about that, and some people think so, some people don't. The duration, of course, is also important. And the severity, the degree of exposure is important. But I'd like to introduce you to a type of adversity that has not been thought of before until we came into the field, and that is really important. It's important because we could do something really good about it, and that's unpredictability of signals from the world. Okay, so we're now going to go into the developing brain. Think about it. In the first 6 months, 1 year, 2 years of life, it makes no difference if you grew up in a small village in Ethiopia or in Beverly Hills. Your world, your environment is the proximate environment and the influence. Don't worry about it. The influence, the sensory input, the signals that you get from your caretakers I apologize to all the men in the room. I'm showing here a mother, but it's absolutely the same thing for father or grandmother, whoever takes care of the child. This is the world, and the signals from the parents and the family and the immediate environment both buffer and filter and convey the rest of the world. And that is nothing new. There's a whole 100 years worth of a discussion of the valence of interaction between, say, a mother and a child, sensitivity, responsivity, whatever. But we need to realize that that also allows stress to get into the developing brain in two ways. One is directly, through hunger, through cold, through beating, if you will. And one is indirect, through a change in the signals that the stressed or depressed parent or caregiver will send into the child. So we realize that we need to look at that also in an experimental animal, because we need to see which is the more important one. Again, when we look at the direct effects, which is what we looked with the ACEs in the scales that I showed you, we got good results in population level, but not individual. Could it be that there's a change in the behaviors of the caretaker? So we went into the cages and looked at behaviors of the mother rat or mouse any way we could. And again, this is a really boring, complicated slide, and the only important thing is that there's no difference between the blue dots and the purple dots. They look exactly the same. And that was miserable because I had no explanation. We excluded that the little pups in the cage, they were not too cold, they got plenty of food, and if maternal behavior is no difference, then what's going on here? I was ready to take my hair out, and that's not very good because I don't have very much. But anyway, that was all the case until we received a video from collaborators. This paradigm, this model of early life adversity, is now used by at least 300 labs around the world, probably more. And that was the video they sent us. You can see here maternal behavior in the bottom cage, which is a regular cage, and the top cage, which is a limited bedding and nesting. Can you see a difference in maternal behavior? Is there anybody in the room who can't see a difference in maternal behavior? All right, eventually that mother mouse will settle down, and the overall, if you quantify her care to the pups, it's the same. But clearly, the duration of each one, it is totally fragmented. It's tiny little bouts of care. In addition, when we began to work with Hal Stern, who's now a friendly provost, and looked at things mathematically and computationally, we realized that the sequence of events is screwed up. Excuse my language. So in a typical cage, you can see that behavior O will follow behavior C in this probability matrix almost 100% of the time, because this is a probability here, okay? In contrast, behavior NB will never follow behavior N. Probability here is 0. So some things are predictable. You can figure out that behavior A will follow behavior B and whatnot. In the limited bedding and nesting cage, everything looks blue and light blue and green. Everything has a probability of 0.2, 0.3, which means nothing can be deduced in terms of figuring out what's going to happen. This can be quantified mathematically as an entropy rate, which we did, or Hal did, and then we can really see that there is a whole lack of predictability, completely chaos. So what appears to be, from going back into the cage, and as I'll show you in a minute in kids, unpredictable and fragmented maternal signals seem to be the driver of ELA-induced plasticity in the brain, changes in the brain, permanent difference in behavior. And how might that happen? How would that be the case? Well, that was discovered quite some time ago. For sensory system, for the develop of the visual system, for the develop of the auditory system, okay, those circuits that go all the way from the eyes and the ears to our cortex, to our brain centers, that's been well known. For example, for the visual system, that's been nicely shown that if you go ahead and block light and pattern of light from one eye of a kitten by patching it, two things happen. One, you get the Nobel Prize, because this is what Hubel and Wiesel did. Second, that kitten will never see right. Takao Hensch was able to show at Harvard that for the sensory system, you need to have many different sounds in many different patterns to actually develop a normal auditory system. Otherwise, you don't. And there's a sensitive period in which that happens. The French have done the same thing with the somatosensory system, and we posit that that may be the case also for memory circuit, and actually for higher complex circuits like those that drive our emotion. And we think that may be what's going on. Now, we've gone into a whirlwind tour from human questions into mice and rats. And the discoveries, hopefully, are interesting. But with all due respect, the mental health of mice and rats is just not something that's the most important thing in the world. Okay? So the question is this important in children? And that required really a large, amazing group of people to come work together. We brought together people from UCI and also from UC San Diego and Denver and Chapman. And these people have remarkable expertise in imaging, statistics, psychiatry, psychology, bioinformatics, epigenetics. And this was really what is the Conti Center. And we really set out to ask, is this important in children? Now, why do we care? Well, there are many different kinds of adversities, as we've just gone over them, and we would really love to be able to prevent all poverty, neglect, abuse, war, migration, what have you. And I hope that one day we will. But for unpredictability, we can teach parents. We can teach folks. We can really, this is tractable, addressable. And if it really contributes to outcome, which is what we found in mice and rats, that will be important. So we tackled this question, and the first paper that came out in the prestigious Proceeding of National Academy of Sciences showed that, yep, exposure to unpredictable maternal sensory signal in the mice and rats, it's either fragmentation or predictability. In the human, it's really unpredictability, really influences cognitive development in kids. So again, in rats, it was easy. The low predictable cage was the limited bedding and nesting, and I've shown you how the mother behaves. And the high predictability was another cage. We don't put kids in cages, so you have here a spectrum, a scope. But as you can see that the more unpredictable maternal behavior, the less well the kid will do on delayed recall, which is a hippocampal function, okay? This was true even when we did statistical models and realized that everything that we know about other kinds of adversity, that doesn't really change that. Maternal sensitivity, age, education, depressive symptoms, whatever. That has now been replicated by several different labs, not only in the United States, but also in places like Finland and other places in Europe. So it's real. So how did we measure unpredictable maternal behavior? Well, we started off the tough way, in which we looked at what the mother signals to the child, or father for that matter, auditory, sound, talking, tactile, touch, and visual, looking at it. And we had to throw away all the valence in terms of is it good or bad. Just look at this, at the signals. And this is what I've shown you in mice and rats, now done in kids. Again, you can get a probability matrix, although a state matrix, and this showed you the 8 possibilities of one alone, whatever, and you can see if they're predictable or unpredictable, and again, calculate an entropy rate. Well, that would work in a research setting when you have a bunch of undergrads that can score videos, or maybe we can teach AI to do that. But this is not something that we can really scale up or really answer important questions about what goes on in the world. So the group, led by Laura Glynn, developed a questionnaire. And that questionnaire is called QUIC, Questionnaire of Unpredictability in Childhood. That can then go into clinics, into laboratories. You can ask children and parents, you can ask adolescents, you can actually ask adults what their childhood was like and see if you can correlate that with what's going on now. And this has sort of taken the world by storm. It's now, to my knowledge, translated to 11 languages, including Arabic, Hebrew, Finnish, Dutch, German, 2 different kinds of Spanish, and what have you. So that was good. But that really wasn't good enough because there are 38 questions, and again, people are busy. So we distilled that into really 5 questions that get of the essence of the environment of a child is really predictable, routine, or unpredictable. And that is a 5-item instrument. And then we were ready to go. So let me recap what we've done here in the human study. First, we really needed to know if if early life adversity really causally leads to problems later in life. We needed an animal model that shows us causality and some mechanism. And then that animal model really uncovered this new type of adversity that we didn't know about, unpredictability. We then needed to figure out, well, does that work in children? And we had to create instruments, such as the original entropy measures and then QUIC and QUIC-5, to really test this in children. And sure enough, at least in experimental control trials, we found that unpredictability is important. It adds to other adversities and really changes the brain. We then also asked, working together with the Marine Resiliency Group in San Diego, if unpredictability is important for adult mental health. And yes, it turned out that unpredictability generates anhedonia in veterans and in recruitees, as well as in the mice. And that then really governs to a degree who's going to come back with Afghanistan or Iraq with PTSD. But then we really needed to go ahead and scale that up to really ask the many different adversities here in Orange County, California. Is unpredictability important in Orange County, California? And that's really what I'd like to end by telling you. This is the final story. So having the Quick 5, we were fortunate and privileged to get a grant from the state of California on precision medicine, because our goal is to go from the population level to be able to predict for an individual child what is going to happen. We teamed up with CHOC, or RadioCHOC, with the Chapman University, and went to ask, can we really find if in the amazing spectrum, in the broad representation here of kids in Orange County, is unpredictability important as a predictor of how they will do as adolescents and adults? And we rolled this in 19 primary care clinics here in Orange County, anywhere from Clínica para los Niños, which is not a particularly well-off part of town in Santa Ana, all the way to San Clemente and Newport Beach and what have you. And we've now gotten questionnaires in more than 70,000 kids, and what I'm going to show you is the analysis of 30,000 kids here in Orange County, California. And the question was, pediatricians in California are mandated by the state to actually ask about adverse childhood events, to run that scale of adversity that I showed you at the beginning, okay? They call it a PEARLS here. And we were able to add the Quick 5, which is this 2-minute, just 5-item scale that asks about predictability and unpredictability. And here are the results to start off with. So the PEARLS, the regular scale that we told you, sure enough, the more problems you had, the more adversity you had, a higher probability of having anxiety, HDD, depression, and in kids, externalizing problems oftentimes lead to other mental health problems and sleep. The Quick 5 was just as good. So you can say, now wait a minute, if I'm getting all that information with the regular scale, who needs the Quick 5? So then we had them neck to neck, and this is what we really found. Now this is complicated, so I'm going to walk you through that because it is really quite remarkable. Here we have the PEARLS score. This is a scale that we all know, okay? And surprise, surprise, compared to a kid that has no adversity, if a kid has 4 different ones, the probability of getting depression as an adolescent is 13-fold. But that was known. The other people have shown that in inner city Atlanta. But now let's look at kids who don't have any known adversity. Nobody will ever pick those kids up. But see what happens when they have a lot of unpredictability in their life. Their probability of getting depression is practically as high. These kids would never be detected any other way. This is really one of the sources of outcome prediction that we never knew about, and now we do. And again, unpredictability is tractable, actionable. So that is where we are now. We've looked a little bit at the question. We understood how the brain encodes early life experiences and how they get transformed later in life. And we also know that some of these research discoveries that we find in animals, both in terms of mechanism and how things work, really can be helpful. So where do we go from here? Well, we got a long way to go, because I think we have a really, really, really important clinical question, clinical problem. How do events that happen to us early in life influence us? If we know the how, if we know the mechanism, if we know the molecular events, we can actually design medicines. And this is why we need more preclinical animal studies. There is no way that we can rely on existing information if the information does not exist. We really need to do much more using novel technology to really come up with medications that we can treat, or with ways to prevent, or alter the plasticity, or change the sensitive period, all of which are really tractable questions. We then need to go and validate whatever we find in mice and rats in humans. And I've shown you a way to do that. And that needs to be done concretely. It requires a fair amount of resources to really do this right. Once we know that we have a pathway, then we really need to go beyond that into the community, into policy, into intervention. So there's a huge amount of stuff that we need to do, but we can and should really address this type of important clinical questions. And I'm going to stop here and really thank the folks in my lab, without which this would never happen. And of course, all the collaborators here at UCI, with the Connie Center, and pretty much throughout the world, and the people who have been able to fund this and make the whole thing happen. And also you for listening. Thank you.
Thank you, Tallie. That was an amazing tour de force performance of translational science at the heart, from bench to bedside. So I'm sure there are a lot of questions from people in the audience, so why don't we open it up? And I think someone's running around with microphones. So Craig, you're the first.
Tallie, beautiful talk. Just a quick question about, is this early life events having this kind of later effect on adult life, is that something you think is evolutionarily conserved, or is it just aberrant development?
It's very much evolutionarily conserved. I mean, the idea of early life connection, I think started with Conrad Lorenz somewhere in the 1920s, and probably before that in terms of study. But maternal behavior starts actually even in insects. If you go look at like planet Earth and David Attenborough, and you can really see this throughout species. So yeah, there is something called imprinting, and we now know that some of the imprinting is epigenetic changes. The changes in circuitry and whatnot is pretty much in, I wouldn't say all multicellular organism, but definitely in mammals and probably before that. Actually, you see it in birds as well. So it's very evolutionarily conserved. The peptide CRH, by the way, which is the stress sensor, is almost identical in toads and frogs. So this is, I mean, these principles are very, very, very evolutionarily conserved. It's a wonderful question. Thank you.
Dan.
Great talk. Thank you. One of the things that strikes me is that people, when they grow up to become parents, often pattern their parenting on what their own parents did. So it strikes me that what you've laid out here is sort of a way for multi-generational perpetuation of these kind of behaviors, and children fall further and further behind.
I assume you'd like for me to comment on that. I think you're absolutely on. There are a number of groups around the world that are actually looking at that, and some recent papers are done. We haven't yet, because there's just too much to do, though, because it's not important. But there are groups, both in France and in this country, who've actually gone 3 generations and suggest that what you have here is real. So again, the way to mitigate that, the way to avoid this falling behind, as you say, is really, number 1, to be aware of it. Number 2, to really see about educating, about explaining, and maybe break the chain. Yeah, absolutely.
Tallie, thank you for a beautiful talk. It was very informative. One of the questions I have is, in terms of adversity in human populations, do we understand the breadth of what adversity is? And one of the things I'm thinking about at the moment, for example, is, do children in Hispanic households watching the news, are those traumatic events for them, for example, when they see children being deported out of the country? And then the follow-up to that is, in terms of your work of parental behavior, are there ways that you think, again, in human populations, parents might behave that would either exacerbate or alleviate some of those adverse reactions?
Now, Peter, this is a classical Magnificent Peter question, because you go smack into the heart of things. let's put it aside, let's not talk about Hispanic children being traumatized. I think you could talk about regular adults being traumatized by some of these pictures. So I think you're right on. There is a very large body of literature discussing modulation. The idea really is that, and this is related to predictability and otherwise, yes, there are things that every child in 2025 and 2026 and other time are exposed to. And there's really good evidence that family can modulate that. In fact, that's the other factor that probably, aside from genetics also, that really accounts for the mismatch between ACEs and outcome. Again, genetics is one, and predictability is one, and there are probably others. We looked at family predictability in general as a potential way of doing it, namely that, yes, the world is falling apart, but mommy and daddy are here for me and they're there routinely. We actually had a natural experiment that we did, again, led by Laura Glynn in COVID, because here we are trying to look at unpredictability, and this 800-pound gorilla, COVID, showed up. How do we handle that? So we very quickly sent out questionnaires and got them from several hundred kids. And the bottom line is that, yes, with the complete disruption with the pandemic, was not good for mental health. We look at preschoolers there, okay? They were just miserable. I mean, extreme behavior, externalizing, whatever, you name it. But the families that the kids were more or less protected were not distinguished by socioeconomic level, were not distinguished by ethnicity, where they were distinguished by the fact the family kept their routines. And that paper has made enough noise that the Surgeon General of the United States at that time actually cites it. So the short answer to your question is absolutely. The family really needs to be consistent and predictable, if you will. So thank you.
Front? Yeah.
How do you assess predictability, or what are the 5 quick questions?
I think I actually have them here. So maybe I'll just take the liberty of showing them. And again, you can ask that a child themselves, or an adolescent, or even an adult. Or they can also, before the age of 9 or 10, we ask the parents. And these are the questions, okay? So my child has bedtime routine. At least one of my child's parents is completely unpredictable and actually can go from pleasant to absolutely mad within seconds. My child's parents have a stable relationship. And in my child's house, things that we need are often misplaced and what have you, okay? So these are very, very simple questions. And the way to actually educate is really to build that for each family. Because in some family, it's a morning routine. In some family, it's an evening routine. The idea is to really have routines and predictions. Thank you for the question. Yeah, it's
Okay. Yeah.
Like the very field of psychotherapy, going back to Freud and then Jung, and then the way that field is continued is based, a lot of it is based on understanding childhood trauma and using that to treat the patient, and even modern psychotherapy sometimes can address that. Do you feel that conclusions that are being made from the neuroscientific perspective on the effect of one's childhood, like neurologically, can be used in order to help current clinicians gain better patient outcomes, and for even people on their own to kind of navigate and deal with the effects of their own childhood?
So the question was about psychotherapy and the theories of Freud and Jung in terms of how to think about childhood trauma and go on from there. And the question is, can neuroscience help that? I think it's a really, really good question, and it really looks at the world in very different perspectives. So, you know, I'm not going to change a Freudian psychiatrist view of the world. They just think about that. What we are thinking based on neuroscience is what we as adults believe may or may not need to be traumatic, may or may not be. That I think we really need to understand the fundamental things that happen in an individual's brain, and what's traumatic for one person may not be for the other. And I think if we understand the mechanism by which early life adversity influences the brain, we will be better off. And I've shown you scenarios here where early life adversity has adverse outcomes. I think to some degree, depending on the duration, depending on the support from the family, depending on other things, it may actually build resilience. And we're actually using the epigenetics also to look at what are the resilience programs. So I think your question is a good one, but I don't know that psychotherapy will be the manner in which we will employ or deploy interventions after the information that we get here. I may choose other tools, education one of them.
More questions? Yep.
For context, I lecture in criminology, law, and society, particularly in juvenile delinquency.
Yeah, just for context, I lecture in, as I said, criminology, law, and society, juvenile delinquency. So your answer to that last question was where I was going. Because when you were talking about where we go from here, you mentioned maybe the development of additional medicines. But didn't hear you talk about other types of interventions where this research might lead us to.
That's a very good point, so I apologize because I definitely do mean that. I think we need education, I think we need policy changes, I think we need to go to different communities and tailor intervention such as education and other types of support into the specific ethnic and I would say more gestalt type ethos of each community. And that is, I think, the beauty of what we're doing here, because the study that we did, the community advisory board that we have in looking at that, really is representative of Orange County, California, and of California in general, with people from all strata of life and people from many, many different backgrounds and countries, so that we can actually, number one, have a broad understanding, number two, allow precision medicine. And medicine is not necessarily molecules and drugs. Allow precision intervention that's appropriate for each family. As we mentioned a little while ago, for some families, routine may be very different than from other families. It doesn't make a shred of a difference as long as it is a routine. So you're right on. It's not necessarily only new drugs that we need to do. Much can be, I think, or should be thought about prevention versus intervention and therapy. Because I think the approach to a family with a 2-year-old will be very different than to a 50-year-old who now thinks that something bad happened to them. And in this particular case, it is conceivable that an intervention with medications that do something to specific circuits might do something. But I think you're absolutely right. So thank you.
Okay, let's take the last 2 questions here. Okay, Dennis?
So I'm not playing on words, but I'm thinking about inconsistent consistency. The raising of children is made up of 1, 2, 3, 4, 5 people, and within that pool, you may, to simplify it, you may have a father who is inconsistent and a mother is consistent, or you might have a consistent parents but have uncles or other people that are involved that are inconsistent. So it seems to me it kind of becomes a jumble, and I'm curious about, would one have a stronger influence than another? Otherwise, consistency over inconsistency.
Yeah. It's a really good question, and I can't really answer it appropriately because we also need to include age. Think about it. In the first 2 years of life, which is why I chose that baby, essentially the world of a child is a parent and the immediate family. This is, if somebody here knew theory, it's Piaget, right? When you begin to be 3, 4, and 5, the influence of parents becomes less and less and less. And it's the other community that becomes important. And again, in other questionnaires, we also look at that. We can have a really consistent family, but if you hear gunfire, 3 times a week, that is pretty inconsistent and scary. So I think it is a very complex thing to do, and absolutely. I do not think that we have a really easy answer. I think we need to understand, first of all, or agree or not agree, that we have some new things that we now know influence the brain early in life, that influence our life later, and then think as individuals and as a society how to best handle them. So your question is right on, and I wish I had a simple answer, but I really don't. So thank you.
Okay, let's do one final question.
Thank you, Dean. I get the last question. I just want clarity on this as a clinician. When you say my child at bedtime, was this for children 5 and under, or what was the age range of the Quick 5?
Yeah. So the Quick 5.
Did I miss that?
Yeah. First of all, this is a wonderful question. What kind of clinician are you?
I actually take care of geriatrics, but I come from,
Absolutely.
but it's a circle of life, right? But
absolutely.
I actually represent the Vietnamese community steeped in trauma from
Beautiful.
war, and
Yeah.
it's a personal as well as a professional question.
Thank you so much. I really appreciate that. So, and you're right. I mean, it spans our lifetime. The questionnaire can be tailored. What we deployed in Orange County was really addressed to 10 and below. That was the one that you saw because I showed you the parent version. And for 10 and above, we have a version that is actually to the adolescent, to the child. And we also have that for adults. So the questionnaire comes in two versions, English, Spanish, and hopefully Vietnamese very soon. One is addressed to the child and one is addressed to the parent. So what you saw here is really what was addressed to a parent in the clinic where CHOC sees primary care clinics show kids all the way to 18, to my knowledge. Okay? So it's a really astute question. Thank you.
It wasn't just for infants,
No, it's not just for infants. Absolutely not.
In your presentation, you also mentioned that early adverse events in childhood as well as adolescence. So the questionnaire was used until 18, not just 5 and under.
That is correct, but a different version was used 10 and above when we asked the child directly.
Okay. Thank you so much.
Absolutely. And it's available, by the way. It's in that paper. It's available online, available on our website. I would love to see it. I don't think anybody is doing it in geriatrics yet. So I think that would-
Okay, with that, let's thank Dr. Baram again for a truly wonderful and captivating talk. Tallie, we have a little bit of a gift for you to thank you and show our appreciation to you. And lastly, I just want to invite you all. We have a special reception out there. And let me introduce Hazel real fast, because you've heard me say that we are kind of like PBS. We depend on viewers like you. So if you're interested in seeing these talks continue, please talk to Hazel. So we'll see you all out in the reception, and I'm sure Dr. Baram will be excited to answer some of your questions.